Okay, today we're going to be looking at observing microbes. So this lecture is going to be based off microscopy. Looking at light microscopy, we have a couple different modes that we'll be getting into throughout this lecture. And beyond light microscopy, we'll even be getting into electron microscopy. So we're going to be comparing and contrasting the different types of microscopy used in microbiology. To start out with, the reason why we would use a microscope is to amplify what we're seeing, to increase something in size. So looking at microbiology, the organisms that we're looking at mostly cannot be seen by the unaided eye, by the naked eye. So using a microscope as a tool to see them is going to enable us to analyze microorganisms from a visual perspective. Looking at a microscope itself, a microscope is actually quite similar to how our eye works. If you see this image at the bottom of the slide, this is how our eye works, and it focuses the image on the retina. Something to take note of is that the image on the back of the retina is actually upside down. So we actually see upside down, then our brain reorients it to be right side up. Well, microscopes, in fact, work the same way. That actually, as we look through the microscope, the image is inverted. It's actually upside down. Now, this isn't really too much of a problem for our understanding of microbes in microscopy because microbes really don't have an up or a down or any direction that they need to be. Okay, so magnification itself is just increasing the apparent size of an image. And resolution is going to be able to define the difference between two objects that we see, 2 points. If we have poor resolution, it could look blurry. We could see 2 points that are close to one another and wouldn't be able to distinguish them. If we have high resolution, those 2 points will be defined two separate points that we see without any blurry or fuzzy vision between them. Looking at the size in microscopy, we know that cell sizes are going to vary quite a bit. We looked at prokaryotic and eukaryotic cells prior to this. Eukaryotic cells are our larger cells that we're looking at. Here we can see trypanosoma, which is a large eukaryotic organism. Then we can go down to a red blood cell. This is a great reference because this is a cell within our body. It's this biconcave donut shape. And something unique about red blood cells is actually there's no nucleus within them. So they are smaller because they don't have all of the same membrane-bound organelles like a nucleus that most eukaryotic cells have. So red blood cell comes in at about 8 microns in size. And underneath that, about half the size of that, we start looking at the larger end of prokaryotic microorganisms. So looking at them, we can probably point out E. coli here, one that we're probably familiar with at 2.5 microns, Staphylococcus aureus at 0.9, and then we even move all the way down to the herpes virus at 0.2 microns. So viruses, we can see, are quite a bit smaller than microorganisms, and microorganisms that are prokaryotic are quite a bit smaller than eukaryotic organisms. So the size of these organisms is going to be relative to what kind of microscopy we're going to be using. In light microscopy, eukaryotic organisms, as they're large, they have membrane-bound organelles, are quite easy to see. Moving down into prokaryotic organisms like archaea, bacteria are quite a bit smaller. We can still visualize them with light microscopy for identification. But if we really want to get more detail, we're going to have to get down to electron microscopy. And when it comes to viruses, most viruses cannot be seen with a light microscope. We would have to have a higher resolution. So we'd move to electron microscopy for visualizing them. Okay, so when we say LM, that acronym represents light microscopy. Using light microscopy, we can stain our samples so we can visualize it a little bit better. If we don't stain our samples, our microorganisms just look clear almost on the slide, unless they're naturally pigmented themselves and maybe have chloroplasts within them or chlorophyll molecules. Moving on to phase contrast microscopes, we can call that PCM. What we typically used is stained samples on here, but you can also use unstained samples. We can move down to fluorescent microscopy, where we will be staining our sample, but the stain is a little bit different. We're actually using a fluorescent dye that is going to emit light that can be picked up, and we'll see that imagery in just a bit. Following that, we have electron microscopy. Electron microscopy is really important because it uses electrons instead of light waves to create a higher resolution. We have SEM, which is scanning electron microscopy, and we have TEM, which is transmission electron microscopy, both which use electrons to amplify the size of the sample. Okay, looking at microbial shapes and sizes, again, we have all different shapes and sizes going from prokaryotic to eukaryotic organisms. Here we can see an amoeba on the left, and we can see these pseudopods. This is an unstained sample on the left, visualized through light microscopy. On the right, we can see a stained microorganism, and that's why it has that purple pigmentation. There's actually a red blood cell there as well. So we have this eukaryotic organism and a red blood cell. So they're quite a bit different in sizes that we're seeing here. Okay. Looking at microbial shape and size, microbes are extremely diverse in their morphology. So we can go over a couple of them here. At the top, what we see is going to be light microscopy. The bottom is the same exact microorganisms, but using electron microscopy, specifically scanning electron microscopy. So here we can see our sample just with light microscopy, and we can see all the more detail with the scanning electron microscope. And that's for all of these samples here, whether we're rod, cocci, or any other form. We're going to see a lot more detail when it comes to electron microscopy. Okay, also with these samples, everything on the top has been stained. So you see pigment that's attached to them that adheres to those cell walls. At the bottom, these are actually not stained sample. These are pseudo-colorized, meaning the color is added after the sample is scanned. Okay, looking at different sizes, we have quite a different size scale. Some microorganisms that are eukaryotic can be seen with the human eye as they're quite large or just the light microscope. Moving down, we have bacteria with the light microscope. Then we can get into transmission electron microscopy. So moving from this sample to this sample, we see with TEM, we see a lot more detail of the Bacillus bacterial sample. Moving on to image D, what we're actually seeing here is transmission electron microscopy, and we're seeing what we call a polysome, which is a RNA, mRNA, with ribosomes attached to it, making proteins. So those little squiggly tails that you're seeing coming off is actually the primary sequence of amino acids for a polypeptide chain being created. And X-ray crystallography, as we see down here, can actually give us the ribosome structure. Okay. Looking at light and how we use light, it's based off of the wavelength or frequency of light. We know the visible spectrum of light is the colors of the rainbow from low frequency to high frequency. And so whenever we're using a light microscope, we're using this range of frequency. As you can see here, X-rays are going to have a higher frequency, and we can continue moving to gamma rays, and on. So we see we can increase the frequency. When we look at magnification, it involves refraction or the movement of light through different types of mediums. Here we have light going through a gaseous medium, so the atmosphere, the air around us. And then it hits the glass. So this could maybe be your cover slip that you're hitting. As it hits the glass, it's going to be refracted. The angle of the light is going to change. Here we see from 60 to 34.5 degrees. And then it moves through at 34.5 degrees, moving through the glass, and then moves back out of the glass, maybe into a different medium. So this could be an oil medium if we're doing oil immersion. It could be a wet mount with water as what we're moving into. But refraction will change as it moves through a different a different medium, and it depends on how high the refractive index is of that medium in how the wavelengths are going to bend. So here we can see a curved surface is a lens, and this is actually how glasses work as well. So we see light coming in, and then it's being refracted. So it changes that angle of movement as it moves through the glass and is focused on a focal point here. So this is like our microscope lens, and the focal point is where they're all going to hit and then reflect again. So very similar to what we saw right here. We have our focal point, and then we have the image on the retina. So a microscope is working similar to how our eye actually works. Okay. When we talk about resolution, again, it's about defining the difference between 2 points. Here we can see 2 points that are barely resolved. So we see poor resolution. These might look blurry, and you wouldn't be able to distinguish the fact that there's 2 points here. On the other hand, we have 2 points that are highly resolved. We see them as 2 separate individual points. So just like on a TV, if we're looking at how many pixels there are per a certain area, this would show you more pixels per area. So you're going to see more defined points, more resolved points in an area. So this is the difference between our low-powered magnification and our high-powered magnification. We're able to get higher resolutions as we move our objective lenses up from something like 4x to 10x to 40x to 100x. We're actually increasing the resolution between parts and seeing greater definition. When it comes to observing microorganisms with light microscopy, we're going to be using stains to visualize them. If we don't use stains, they'll just look kind of clear against a clear background for the most part. So we fix our sample to the slide so that it's not moving, and then we stain them. This process actually ends up killing the cells, so we're not going to be looking at living, mobilized cells at this point. So if you do something like a wet mount, you're looking at live cells and you may see movement. But if you're doing fixation and staining, making a slide prep, a smear, and then staining, you're going to be actually killing the cells and you're going to be adhering them to the slide, normally heat fixing them to the slide, and then follow with a staining procedure. The stains that we add are actually positively charged and they bind to our negatively charged cells. So most microorganisms have a cell wall that contains a negative charge. So positive charges will adhere to the cell wall. Here we see methylene blue and crystal violet. Those are 2 examples of stains that can be used to observe microorganisms. When we call something a simple stain, that means we only use one stain, essentially, and we're staining the microorganism itself, but not the background. Let's go ahead and review the process of staining here. So here we would take a loop full with an inoculation loop, that's our tool in microbiology in the laboratory, to grab some of our sample, and we would place that sample on a slide for observation. Then we would spread it out to create a thin layer. This is quite important for a couple different reasons. We want a thin layer so it can air dry quickly, but we also want a thin layer because these microscopic organisms, if coming from a high concentration culture with high biomass, will end up with cells piled on top of one another, and you won't be able to see much. So by spreading them out, you're hoping they're going to be able to see individual cells and really observe their morphology. The samples air dry. Once the samples are air dried, or there's other modes of drying as well, methanol, we have to fix the sample to the slide. So sometimes we heat fix it as well. Then we can go ahead and look into staining our sample. So here we see they're using methylene blue. So they're adding this blue pigment to the cells on the slide. And they're going to wait approximately a minute, depending on what you're staining. This can change. So they have methylene blue sitting on the slide for a minute. It's being essentially absorbed by the cells. It's adhering to that negatively charged cell as it's a positively charged stain. Then we go ahead and wash off the stain. So that's just water rinsing off the stain. And then we blot it with our bulbous paper in order to remove any excess water from the slide. So now essentially we have our smear sample of the microorganism with a dye on top of it. And that's why we see that blue pigmentation. So that's what we want to line up right underneath the objective lenses of the microscope so that we're able to observe our stains. Another form of stain is called a differential stain, and it's a very similar procedure to what we see here, except for we add another stain. So after we wash off this stain, we can add what we call a counter stain. So we would start again, adding another stain, leaving it off, then rinsing it off. Then we would blot it and observe it. We use counter stains to be able to identify different types of microorganisms. So some may die with some stains, some may die with other stains. So it's a great way of visualizing samples like the endospore stain, the gram stain is seen here where gram-positive cells are going to stain purple, our gram-negative cells are going to stain pink. So we can differentiate between them based off of cell physiology. Let's go ahead and dive into the Gram stain. So Gram was actually the scientist who created the procedure, and he created this procedure to differentiate between gram-positive cells and gram-negative cells. You might ask yourself, why would we care about gram-negative or gram-positive cells? But it's actually really clinically important. So when people end up in the hospital with an infection and an antibiotic needs to be administered for a bacterial infection, we have to differentiate whether the infection is gram-positive or gram-negative. And what that has to do with is the cell wall and membrane structure. So the outer portion of the bacteria. A lot of antibiotics work by targeting the outside of the cell. And if the outside of the cell is a cell wall versus a membrane, it's going to affect how these antibiotics are going to work. It may render an antibiotic completely useless if we're using one that targets a gram-positive cell and treating on a gram-negative. It wouldn't work. So clinically, it's extremely important to know whether a sample is gram-negative or gram-positive. And all cells essentially fall into this gram-negative or gram-positive categorization. There's a couple minor exceptions where the gram stain doesn't work well, but most all cells fall within this categorization. So it's a really easy way to do a diagnostic approach and to start clinical treatment. Let's go ahead and walk through the steps on a gram stain. So let's go ahead and start out with our cells. We're going to run these in parallel. So it would be like if we had two slides, as we see here, and we're running this experiment in parallel. So on our first slide, we added our smear, our sample of gram positive. And on the other one, we're adding our sample that's gram negative. As you see, they're clear. Again, that's what they're basically going to look like. These samples are not pink and purple naturally. That is the stain that's deposited on them. After we add the sample, we air dry it, maybe heat fix it. Then we can go ahead and add our primary stain, our initial stain, and this is going to be crystal violet. Crystal violet has a purple pigmentation. It's left on for about 1 minute to saturate the sample. Then we actually add another reagent called iodine or Gram's iodine, sometimes it's called, which acts as a mordant and really fixes that crystal violet to the cell. So not only do we have that positive negative attraction between crystal violet, which is positively charged, and these cells, which are negatively charged, we have iodine, which is creating a stronger bind between them. So right now, both of our gram positive and both of our gram negative appear to be purple. They seem to be identical. The next step is where we start seeing differentiation between our gram negative and gram positive cells. So we go ahead and wash our sample with ethanol. Ethanol is what we call a decolorizer. So it decolorizes the sample. It pulls out the pigmentation. Here we can see that in the gram-positive cell, the ethanol does not decolorize the sample, but on the gram-negative, we go back to the original pigmentation, which is that clear pigmentation that we saw. Ethanol is only left on for about 20 seconds, so we don't want to overly treat with ethanol. We could wash out some of the dye here. Okay, following that, if we were to look at our sample underneath the microscope at this point, one side would be purple, our gram-positive, the other would be clear, as we see here. Moving forward, we're going to go ahead and add what we call our counter stain. We have safranin as our counter stain, And that's going to add a pink pigmentation. So it's going to adhere to these cells, our gram-negative cells. So our cells are still negatively charged. Safranin is actually positively charged to adhere to it. It can adhere to the gram-positive cells as well, but if we already have purple down and we put a lighter pigment on top of it, like pink, it's not going to really be able to be visualized, and it's not going to adhere as many of that positive-negative interaction is already taken up by the crystal violet that we see. So on the last step, if we want to visualize our sample, our gram-positive cells are going to stain purple, our gram-negative cells stain pink. So that's how we differentiate between cells that are gram-positive or gram-pink, and we could look at an infection and determine what kind of antibiotic would be appropriate based off of the gram stain. Looking at other cells, other types of stains, we have differential stains like the acid-fast stain. The acid-fast stain can be used on Mycobacterium tuberculosis. This is the microorganism that causes tuberculosis. We can take what we call a sputum sample, something that is coughed up from the lungs, and we can analyze it for the presence of these microorganisms. These microorganisms, Mycobacterium tuberculosis, actually have a waxy layer on them. And this waxy layer prevents the stain from actually adhering. So with an acid-fast stain, we use a primary stain and a counter stain in order to identify our cells. And here we can see it with this pink stain here, identifying our Mycobacterium tuberculosis. Because of this mycolic acid, this waxy outer layer on these acid-fast cells, we need to actually perform this acid-fast stain because a gram stain would not give us the right information because that wax would essentially repel these initial stains, and we would get undesirable results from a gram stain that doesn't lead to identification. So if step one was a gram stain on a sample and it came back not differentiated, we could look into doing an acid-fast stain. Okay, let's look into different types of microscopy. So we've mostly been talking about light microscopy using wavelengths of light to increase the resolution on samples. But if we really want to up the amount of resolution that we have, we can use electron microscopy. And what this does is it sends electrons into the sample instead of wavelengths into the sample and creates even higher resolution. We have two forms of electron microscopy to discuss, TEM and SEM. TEM stands for transmission electron microscopy. This top image you see here, TEM, is where we can actually see quite a bit of detail. So here we see ribosomes within a cell, and here we see HIV and even the viral RNA. So we're looking at the macromolecules, nucleic acid like RNA. We're looking at proteins like ribosomes, ribosomes are part protein, part nucleic acid here. And with transmission electron microscopy, you actually can see through the cell. It's transmitting through the cell, almost more like an X-ray or something. So we see this inner detail. If we were doing scanning electron microscopy, as we see down here, we would get a very detailed view of the surface, not the inside of the cell. So scanning electron microscopy would not enable the observation of the viral RNA or the ribosomes that are inside this cell or inside this virus. Scanning electron microscopy is exactly as it says. It's essentially scanning over the sample and giving us an extremely high resolution 3D image of the surface of the cell. And this is a colorized image. Most of the time you see scanning electron microscopy, SEM, it's pseudo-colorized, as seen here. In green is the bacteria. And then this is the trophic tissue type around it. Over here with TEMs, sometimes they're colorized, sometimes they're not, but most of the time SEM is colorized. So you may even see this picture in a different textbook somewhere else, and maybe the bacteria is purple and maybe the tissue type around it is pink because the color is added in after. Okay, this brings us to our questions. Question number one is, which term refers to the lens's ability to produce clear, non-blurry images of a microscopic object? So this again is where you would pause the video to reflect and choose an answer. We'll go ahead and move on to the answer. The answer for question 1 is resolution. Resolution is the term that refers to a lens's ability to produce clear, non-blurry images of a microscopic object. So again, resolution is defining 2 points from one another, whether they look blurry or whether they look separate and isolated. Question number 2, hypothesize what would you observe if you forgot the counterstain step when staining a gram-negative bacterial cell? Okay, and on to the answer. If you skipped the counterstain step, the cells would be colorless and difficult to distinguish. So we could go back to this gram stain slide here. So if we forgot the safranin here, we're going to see the image without that pink pigmentation. It would just remain clear. Okay, moving on to question 3. Deduce why viral particles cannot be viewed using a compound light microscope. The correct answer for question number 3 is because viral particles are too small to be resolved with a light microscope. Yeah, so when we go back to those images in the beginning, looking at eukaryotic cells to prokaryotic cells, all the way down to our non-living viruses, they are too small to be seen with a light microscope. We don't have enough resolution to observe them. Okay, this brings us to our section objectives. So these slides here are meant to be your study guide to fill out promptly following the lecture. These can be reviewed in groups or independently. And that brings us to the end of the lecture.